Hello, and welcome to the Time Lab, where we study the tumor immune microenvironment and mechanics. My name is Meenal Datta, and I'm an assistant professor in the Department of Aerospace and Mechanical Engineering, as well as the Bioengineering and Material Science and Engineering PhD programs here at the University of Notre Dame. In our lab, we use cell, tissue, animal, and computational models to understand the interplay between tumor mechanics and the immune system in very difficult to treat cancers, including those that affect the brain. By studying these biophysical interactions, we can inform novel treatment approaches that could improve survival outcomes for cancer patients. Our researchers include undergraduate students, graduate students, postdoctoral fellows, and staff scientists, all working together towards this common goal. Today, you'll get to see inside of our lab and to hear from some of our current students. Welcome and enjoy the tour.
Hi, welcome to the Time Lab. Hi, my name is Rumbach, and I am the Time Lab lab manager, and I'm also the lab animal technologist. So I mainly work in keeping the lab in compliance with RMS, which is Risk Management Services. So I make sure that the lab is safe for the lab fellows to do their research, and then also nothing is a tripping hazard and such. I also order supplies for the lab and for the lab fellows. In addition to that, I do animal work in another facility, not here in the lab. So we do cranial window implantation, compression cranial window implantation as well, and then drilling burr holes, and this is on a mouse model. Let's go this way and see the rest of the fellows.
Oh, hello. My name's Julian. I'm a third-year graduate student here at the Time Lab. Come follow me while I put some media in the water bath. Now we're just going to wait for this media to heat up. Alright, now that our media is all warm, let's go ahead and feed our cells. So one thing you should know at the Time Lab is that we work a lot with cells. And what that means is that they're our family, so we need to make sure that they're healthy and that we let them grow and we nourish them into these beautiful cells. Here we have 2 BSCs for us to do our cell culture work and 4 state-of-the-art incubators. Alright, so let's grab our cells. We work with a bunch of different kinds of cells, including fibroblasts, immune cells like macrophages and T cells, cancer cells like breast cancer cells and brain cancer cells. You name it, we probably work with it. Let's take a look at some fibroblasts. So as part of my project here at the Time Lab, I'm interested in looking at the role of mechanical forces on breast cancer development, especially in regards to the immunological response and the stromal response. So let's go ahead and feed these cells. Alright, so in order to evaluate the role of mechanical forces on cells, what we use is something called a transwell compression system. So here we have a transwell, and what we do is we seed the cells from this flask into this transwell membrane. Then, in order to study the effects of compression, we apply this force, this compressive force on the cells using this 3D printed weight we created here at the Time Lab. After a specified amount of time, we collect these cells and we do a number of studies including PCR, bulk RNA sequencing, immunohistochemistry, immunofluorescence staining, etc., in order to see the role of compression on immune cell or stromal cell phenotype and function.
Hi, my name is Alice. I'm a third-year graduate student in the Time Lab, and I work on glioblastoma and the immune system and the role of mechanical stress in disease progression and treatment. So here we have our stereotactic frame where we can precisely implant tumor cells into a mouse brain. This way we can study in vivo immune interactions and solid stress as the tumor grows. At the conclusion of a live animal experiment, we can harvest tissues and do exciting experiments such as with what we have over here. This is our lab's tissue slicer. It's called a compressed tome, and it allows us to take precise thickness slices of a living mouse brain for subsequent culture and experiments. So this is one of our unique devices that allows us to get a novel picture of the tumor immune microenvironment.
Hi, my name is Maksym. I'm a second-year graduate student here at timelab, and my project focuses on brain fibroblasts and their role in brain cancer, specifically in modulating resistance to immunotherapy such as immune checkpoint blockade. I'm also assisting with various biocomputational projects in our lab, and to enable these biocomputational projects, our lab has a specially dedicated high-performance computer on the second floor, as well as a 10x Chromium instrument that enables single-cell genomics experiments.
Thanks for stopping by at the Time Lab. If you like what you heard, feel free to reach out to us. We hope to see you soon.